This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. From the American Society for Microbiology and Microbe TV, this is TWiM, This Week in Microbiology, episode 148, recorded on March 16th, 2017. This episode of TWiM is brought to you by Blue Apron. Blue Apron is the #1 fresh ingredient and recipe delivery service in the country. See what's on the menu this week and get your first 3 meals free with your first order with free shipping by going to blueapron.com/TWiM. Hi, everybody. I'm Vincent Racaniello, and you're listening to TWiM, the podcast that explores unseen life on Earth. Joining me today from Small Things Considered, Elio Schaechter.
Well, how do you do?
Oh, I'm frozen. It is so cold here.
Really? No kidding.
We had a ton of snow this week, and it's frozen, and it looks like it's going to be freezing for another week or two.
Oh, my God.
And, you know, when the snow fell, I thought of you out there in sunny San Diego.
Absolutely.
And I know you spent many years in the snow, so you're not missing it.
No, I'm not.
Also joining us today, he's also on the West Coast from Los Angeles International Airport, Michael Schmidt.
Hello, everyone. It's great when planes work.
Michael, you just flew in, right? And
I just flew in, and the plane got here in record time, but then we had to wait for the gate. And Los Angeles is a beautiful day, but it's a little bit smoggy. So I think they're having their inversion issues like they always do in the winter.
So Michael is dedicated because he knew he wouldn't be able to get to his hotel and make TWiM on time. So he stayed at the airport. He's in the lounge, right?
Yes,
Okay.
So it's quiet.
So
Downstairs, it was a zoo.
Hopefully your Wi-Fi will hold up there. You know, we'll see.
We're praying.
See how it goes.
We're praying.
All right. The American Society for Microbiology wants you to know about the ASM Conference for Undergraduate Educators, the premier event in the field specifically designed for microbiology and biology educators. The 2017 meeting is going to be in Denver, Colorado, July 27th through 30th. You should attend this meeting to participate in the formal and informal conversations that will enhance the work you do every day and shape the future of teaching and learning in the biological sciences. Secure your seat today and submit a poster abstract by April 3rd. All right. You can go to asmq.org to learn more about this terrific meeting. Okay. Today we have 2 amazing stories for your enjoyment. And the first one, Elio is going to tell us all about.
Okay. It's a interesting paper that got a lot of attention in the news. It is called Neanderthal Behavior, Diet, and Disease Inferred from Ancient DNA in Dental Calculus. And it has 31 authors, so I won't read them all. The first one is Laura Weinrich. The last one is Alan Cooper. They are from about 11 countries, all European except for one worker from Australia. And what this is about is the fact that if you look at the remains of humans and humanoids and our cousins, the Neanderthals, there are not many bacteria you find associated with the body, or bacteria or DNA associated with the body, which is useful for analysis because it decomposes, etc. But dental tartar, which is here called dental calculus or plaque, seems to be a good place where enough DNA is retained. So you can analyze what's there. So they infer from the finding of the DNA that the Neanderthal, to cut to the chase, ate meat in the Neanderthals living in Belgium in the steppes, and they ate other things in Spain where they lived in forests. And this includes nuts and berries and mushrooms. Okay, the problem with this is very interesting, and one is fascinated by it. There's no question that we would like to know what our cousins of the past ate. And it's interesting already that the inference that they ate, they had different diets. It tells you that they were not just one kind. They were adaptable, and they could use the food that was found in the areas where they lived. Okay, the technique is to get this plaque and to take it to a very clean lab. So there is a lab in Australia, which is a clean DNA lab an ancient DNA lab. It's called the Australian Centre for Ancient DNA Facility at the University of Adelaide. And this is, just to read you, the rooms are UV treated. They have still air hoods in isolated still air rooms, which are designed to have an ultra low level of background contamination. Okay, so this is what it takes, because otherwise you're looking at current day DNA in the form of contaminants. The DNA they have is very tiny. They're really very small lengths of DNA, and that is normally a problem, but they use special techniques which allow them to analyze such DNA. Okay, so what do they see? Well, they see a lot of bacterial DNA, and some of them are sort of regular oral bacteria. However, interestingly enough, the DNA of the bacterial DNA they find in the Neanderthal is closer to that of chimpanzees than of humans. And you can make anything you want out of that. Anyhow, in the Belgian remains, they found that the meat that was eaten was largely the woolly rhinoceros and some wild sheep. I didn't know that woolly rhinoceros are such a source of meat. Anyhow, so this is fun, and you can, as I say, it's one of the interesting things they found a new Methanobrevibacterium species, which is a new archaeon, which makes it this is 48,000 years ago, it makes it the oldest microbial genome. This is a draft genome, so there's a lot of DNA there. Then they have practically the whole genome of this organism. That makes it the oldest such genome in the bacterial world. Anyhow, so 70 base pairs can buy you a lot of bioinformation if you know what you're doing. Let me talk a little bit about the fact that they divide up from this analysis and other analysis, and this is not the only study along these lines, the fact that humans included, fall into two large groups. One includes ancient farmers, hunters and gatherers, and modern people. And another group, which is different, is forager-gatherers, as opposed to hunter-gatherers. So the hunting thing belongs to a different part of the tree.
And that's where the chimpanzees are. They're effectively forager-gatherers, which is probably one of the best explanations, or the most reasonable explanation, as to why some of their Neanderthal DNA or their plaque DNA was so similar to the chimp diet.
All right. Well, that's interesting. I think there's probably a lot of literature on the derivation of the Neanderthals and whether they are actually closer to chimps than we are is an interesting point. Okay, let's talk about mushrooms.
We knew you were going to get there.
Yeah, well, what the heck, that's my passion, so I'll take advantage of it. But it caught everybody's eyes because the headlines in the newspapers are Neanderthals ate mushrooms. Well, let me tell you, it's a little bit tricky, and I don't want to be a spoil sport because I'm really on the side of Neanderthals eating mushrooms. But here's the problem. The mushrooms they found are, one is called Coprinopsis cinereus. It's a inky cap. If you know what it is, those are mushrooms that deliquesce. They sort of melt, the cap melts away. It's another story that we can cover someday. Anyhow, this is a tiny mushroom, about an inch tall, about a half an inch or 3/4 of an inch across, insignificant in weight, and very, very unlikely to be a food source. The poor Neanderthals would have had to spend days and days to gather enough of this fungus to make a mouthful, essentially, or a dish. So it's very unlikely that that's what that was. The other one is called Schizophilum commune, also known as split gills. And that is a cosmopolitan mushroom which is found on wood. And it is edible. It's eaten in some places. But let me tell you something that a very knowledgeable person by the name of Britt Bunyard wrote as a comment to a post we have today on the subject in our blog. Britt Bunyard, by the way, is the editor of a phenomenal magazine called Fungi, and I strongly recommend this for anybody who's interested in fungi because it's really extraordinarily well written and full of lovely information. Anyhow, Britt says, he points out, as I do in the blog, that both of these fungi are known to have caused disease in humans, usually in immune debilitated people, but you don't know the immune state of the Neanderthals. So it's possible that this was not food, but a pathogen lurking someplace in the body. And in addition, he points out that copriners use almost anything for food, and they could live on the bodies of dead people, so the Neanderthals. So it's possible that this is another thing. So it's not clear that finding, and this is a general problem, because finding DNA in plaque does not mean that it got there as foodstuff.
So it could have gotten there after the person died and was lying.
Could have gotten after the person died. Or let me tell you one thing that Britt points out. This schizophilum is very leathery, and it may, although it's eaten in Southeast Asia. It's uninteresting and not very good. It's very leathery. But he says, I think people just enjoy chewing on them for pleasure. And who knows?
Beef jerky.
It's beef jerky.
Yeah, that's right. Did Neanderthals eat beef jerky?
Well, he says,
I mean,
mushroom jerky.
They have to preserve it. The other thing that Elio touched on is the fact that this is plaque, which is a result of a very complicated biofilm. And so we have to ask the question of Elio, how sticky are fungi to mix biofilms in the oral cavity? Because it just could be stuck there. I mean, it goes back to the hypothesis that he just laid out, that the fungi are good to chew and it passes the time like chewing gum, and you're leaving debris behind, and there may be enough mycelia that have DNA that get encased in the plaque, and as it calcifies down, because the Neanderthals never brushed their teeth, there was no crest. So,
no thing, because if they did, there wouldn't be a paper.
That's right. And so let me ask you this question, Elio. So when we're dug up after the zombie apocalypse and they begin to characterize the plaque associated with our teeth, are they going to find all the processed food that we ate and think, what were humans eating? Pringles. What's the DNA associated with Pringles?
They do have a modern human control in this paper.
Yes.
And the only thing they have is malassezia, the fungi in your skin, right?
Right.
Yeah.
And nothing. They don't have any plants, no fungi, no meats. They have some contaminating tick DNA, apparently.
But, if chewing gum was something living today, or maybe chewing tobacco, I wonder if people who chew tobacco, you wouldn't find some tobacco DNA.
Yeah, maybe.
Well, since I'm at a dental meeting, I'll ask the question.
Yeah, you should. Michael, how does calculus form? Do you have to be alive, or can it form for a while after you're dead?
It can form for a while after you're dead, but most of it requires the input of the carbon source. And so the food web of the oral cavity is extremely complicated. And there's a review that Nature published about in 2010, and I'll dig out that figure for the show notes so you can see this complex food web. And I think that's what we're seeing in the Neanderthals and the chimps and the modern human and all of this variation, and geographically, because we do know you can influence diet based on who's contributing what to the food web of the oral cavity.
How interesting. Well, I would hope that in their examining of Neanderthals, which is probably something that's being done on a large scale, perhaps they can find something like spores of fungi in the intestinal tract. Now, that would be much more convincing. If you found spores in the intestine, the stomach, the intestine, you would have a much better case that they ate mushrooms. But that may not be possible. I don't know.
You're not going to get any intestine from these old
Well,
specimens, right?
if you
Why
thought a
not?
mummy, there have been frozen
The
mummies
mummified. Yeah, they may.
that have been on mountaintops of the Alps. And
Yeah.
remember, I think he was in
Yeah,
Austria, and they thawed him out. And I don't remember if they worked him up for what was in his
they
stomachs. I
did.
remember
They did.
I
It's just
remember
no, they
seeing
have,
something.
and I don't know the result, but they certainly have looked. We ought to look for that paper. There's a paper someplace that talks about
That paper.
analyzing the innards of Otzi is the guy's name.
Yes, right.
Give
right.
him a name.
Yeah.
So these individuals were from caves, which is probably why they're able to get teeth at all. If they were just buried in the ground, it would probably be gone, right?
Yeah.
I suppose. Yeah, I suppose they're mummified.
Mm.
And then Neanderthal remains pop up in the strangest places. So there may be some in glaciers, for all I know.
Yeah.
That would be even better. But I don't know. Maybe there are no 50,000-year-old glaciers that are appropriate for the study. I don't know.
Did you mention that this is the oldest draft microbial genome, 48,000 years old?
I thought I
He did.
did.
Yeah.
But
Yeah, he
didn't
did.
hear it.
Again.
Yeah, that's pretty amazing. Yeah.
yeah, it is. It is. It's remarkable. So this is a good story, but an incomplete one. And tune in because you're going to hear much more about it. There's no question that people are going to pursue this.
That's the value of a molecular chronometer like 16S RNA, because you can tell how old it is just by looking at the mutations in the non-essential parts of that tertiary structure that is 16S ribosomal RNA. And that's what molecular chronometer.
Why is it that we're fascinated, or maybe the press is fascinated, that Neanderthals would eat mushrooms? I would assume they would eat mushrooms. Why not?
That's true. And by the way, certainly, if they ate mushrooms, they didn't eat this one.
Yeah.
I mean, there are great, big, beautiful, edible mushrooms around the same areas. So why would you go for this tiny little thing? I'm sort of surprised that the authors made so much, that this is, the press made so much out of it, because it's not a good indication that they ate mushrooms. But you're right. Why not? It's cheap food.
It's out there, right? Now, Michael, let me ask you, this, they found sequences from sheep, rhinoceros. Does that mean they ate it, or could it be just contaminating?
Well, I doubt. I was thinking whether or not they had the sheep in the cave with them. But the fact that they pulled it out of the plaque, that means it's under a layer of calcium. So the DNA is buried. It's literally in its own sarcophagus. And so that's what leads me to believe it was incomplete digestion of the food that was just in their bad dentition. And I think that's what gives it credence, and it really drives home the fact that why they went to Australia to that ultra clean lab. The folks doing stool transplants today are obsessed trying to understand the microbes that are in the donor stool, and they're obsessed with sequencing strain variation. And they are creating these ultra clean labs because if you think about modern humans and our DNA from the workers could contaminate a sample, and how do you know if it's modern or ancient? And the author should be commended for really going that extra mile to demonstrate beyond a doubt that they made certain that the DNA was truly authentic, because we don't know. DNA is DNA. It hasn't changed in 48,000 years.
Much more. Yeah, I think that's a good point. But the beef that I have is not whether this is Neanderthal DNA or Neanderthal associated DNA, but whether in fact you can tell that this was food stuff. But it points out it could be something that they put in their mouth for other reasons, and it could come later. It's difficult.
Yeah.
But it also adds to the fact that Neanderthals were intelligent, because they knew what they could put into their mouth that wouldn't kill them.
Well, I never doubted that. And the way we're going, they may have been smarter than we are.
Well, hence Pringles.
I mean, they definitely say in the abstract, Neanderthal diet was heavily meat-based and included rhinoceros and sheep. And then
That's in Belgium. It's in Belgium.
In Belgium. And then the others, mushrooms, pine nuts, moss. I mean, they think it's their diet for sure.
Yeah.
Right?
There's also another point we forgot to mention.
Yeah.
They found that they ate laurel, which is a cousin of willows, which contains salicylic acid, which is the active principle of aspirin. And so they make a case that this may have been used therapeutically because they found it in the one case where they could see a tooth abscess.
Yeah.
So the other point, which is a little bit, I must say, less felicitous, is they found penicillium, the fungus penicillium, and they make out of it that they may have been using penicillin, forgetting that most strains of penicillium, most species of penicillium do not make penicillin.
Yeah.
It's one, Penicillium notatum, that does. So finding penicillium does not mean they use penicillin. But certainly it's tantalizing to think that the Neanderthals were evolved sufficiently to be their own doctors. I don't doubt that.
Yeah, it's interesting. Well, I have no doubt that it's interesting to know how people behaved and how they ate a long time ago, for sure, especially Neanderthals, where we don't have a lot of information about them. So this is obviously very appealing. But I think, as Elio said, you have to be careful. Only so much you can get from DNA sequences, right?
That's right.
But, as you get more specimens and more sequencing, some of this might be solidified and some of it not. So I think that'll be really interesting as we go forward. Thank you, Elio.
My pleasure.
Very interesting story. That's it, Michael. That's so cool. And yeah, are they going to find Pringles in our teeth? Wow.
Well, the other fun fact is the Italian version of ancient humanoids actually had the Mediterranean diet. If you look at it. So did the Mediterranean diet come from humans or our ancient cousins?
That's right. Now, speaking of food, I want to remind everyone that we did a paper on TWiM some time ago where they looked for DNA on the keypads of ATM machines here in New York City, and they found lots of food on those keypads. Food DNA, depending on the particular neighborhood, they got different kinds of food. So food is, you can find out what people are eating in many different ways. Pretty cool. This episode is brought to you by Blue Apron. Blue Apron's mission is to make incredible home cooking accessible to everyone while supporting a more sustainable food system, setting the highest standards for ingredients, and building a community of home chefs. We have scientists here who love to cook. It's natural. You follow a protocol. You add things. And you're going to love Blue Apron. And, of course, those of you who are not scientists, you probably like to eat. And if you never were a fan of cooking, this really makes it easy. I don't mind cooking, but I hate getting all the stuff together. And this Blue Apron takes care of it. What they do is deliver seasonal recipes along with fresh, high-quality ingredients so you can make home-cooked meals. You get a box. Each meal comes with step-by-step, easy-to-follow recipe cards and all the ingredients you need, except salt, pepper, and oil. So in 40 minutes or less, you have a great meal. And if you spend a lot eating out or at high-end grocery chains now, you're going to be amazed. Less than $10 a person for healthy home-cooked meals. And the reason they can do that is because they don't have stores at Blue Apron. They have a network of 150 farms, fisheries, and ranchers across the U.S., which they use to get all their ingredients. For example, seafood is sourced under standards developed with the Monterey Bay Aquarium Seafood Watch. Beef, chicken, and pork come from responsibly raised animals, produce from farms that do regenerative farming. And here's the key. They ship you just what you need to make your meals. You can order them 2 people or 4 people at a time. So there's no waste, and you don't overeat. You don't eat too much. You don't go back to have another portion because there isn't any. You've eaten it all up. You can customize your recipes every week based on your dietary preferences, meat or non-meat. You can get a delivery option that suits you. It doesn't have to be every week. And they change their recipes frequently. And within a year, they're not repeated. So you're never going to be bored because you're not going to get the same thing twice. They have amazing recipes. Let's go to the website and see what they have now. I have a few I've been reading for March. But let's see what's on the recipe for today. Ooh, look at this. Beautiful pictures. Top Chef spice-rubbed pork with sweet onion and black beans. Chicken meatballs and fregola sarda with kale and Sicilian tomato sauce. Katsu-style catfish, with the breading on it. Black garlic mayonnaise and jasmine rice. green garlic pesto pasta with butter lettuce salad and creamy lemon dressing. Sorry. Oh my gosh, this is so good looking. Even if you don't want to listen to me, go to blueapron.com/pages/sample-recipes and scroll there. Oh, you're going to see these great pictures of the recipes. Check out this week's menu and get 3 meals free with your first purchase with free shipping. Go to blueapron.com/TWiM. blueapron.com/TWiM. You'll love how good it feels and tastes to create incredible home-cooked meals with Blue Apron. So don't wait. Blue Apron, a better way to cook. Just had lunch and I'm starving. It's blueapron.com/TWiM. All right, we have a paper for you today, which was recently published as a letter in Nature. The title is New CRISPR-Cas Systems from Uncultivated Microbes. And the authors are Burstein, Harrington, Strutt, Probst, Antharaman, Thomas, Doudna, and Banfield. Well, these authors are from the University of California, Berkeley, Lawrence Berkeley National Laboratories. And here we need to talk a little bit about CRISPR-Cas, just as a background. Because in this paper, what they do is to use metagenomic data sets from uncultured microbes, just from environmental samples, to try and answer the question, can we identify new CRISPR-Cas systems? Now, CRISPRs, that stands for clustered, interspersed, short, palindromic repeats, CRISPR. They provide microbes with immunity to foreign DNA, and that includes bacteriophage infection, conjugation, plasmids coming in, transformation by foreign nucleic acids. Any nucleic acids, DNA or RNA, that come into the cell, they encounter the CRISPR system. They're found in about 40% of sequenced bacterial genomes and 90% of sequenced archaeal genomes. Now, CRISPR-Cas is a kind of acquired immunity. They're basically segments of DNA with short, repetitive base sequences, and these sequences are interspersed with spacer DNA, and the spacer DNA is from previous exposure to foreign DNA. And right next to these arrays, these CRISPR arrays, are small clusters of genes. They're called Cas genes, CRISPR-associated systems, and they're involved in the way these things function. So basically, you have these arrays, usually 50 or so units of the repeat spacer sequence in an array, and there can be multiple in a bacterial genome. And again, these are all little histories of the DNA that have come into the cell. By the way, the repeats are about 28 to 37 base pairs in length, and the spacers, which come from the foreign DNA, they're typically 32 to 38 base pairs. Right. So what happens is RNA is transcribed from the spacer sequence. It starts at the beginning, and this is a long transcript that encodes all of the repeat spacer sequences. It's then chopped up by Cas proteins into the individual sequences that'll then target the incoming DNA. And there are a couple of different Cas genes that are involved in this. So there are Cas genes that are involved in acquiring the DNA and helping to put it in the genome. There are Cas genes that are involved in processing the CRISPR RNA transcript. And then there are Cas genes that are involved in degrading the incoming DNA using the CRISPR RNA as a guide. And these have been divided into two classes with many different types. But basically, class 1, the Cas system is a multi-protein array, and then class 2, the Cas is a single protein like Cas9, which binds the RNA and directs it to an incoming DNA. Now, the big news about CRISPR, of course, is that a simplified version has been modified so that you can do genome editing. It's called CRISPR-Cas9, at least one of them. And Cas9 is the nuclease, and you make a synthetic guide RNA. These systems often have 2 different RNAs that are needed for targeting. What they have done in this synthetic system is to make that into one. So you have a target RNA, which you can put in a cell, and then the Cas9 will cut whatever sequence in the genome that you've told it to cut. So you could use 2 sequences separately and excise a gene. And when you do cut out a gene, the ends get repaired together by the cell. Or you can cut out many, many things at once. I just read a paper where someone, Elio, you'll like this. someone removed 16 genes from a fungus all at once to try and modify it for agricultural purposes. So you can do multiple editings all at once. You simply introduce the Cas9 protein and all of the synthetic guides that you want into the cell. Okay, so back to CRISPR. A few more bits of information that'll help us with this paper. Class 1 systems use multiple Cas proteins. As I said, Class 2 use a single large Cas protein like Cas9. And many organisms have multiple CRISPR-Cas systems of different sorts. So what they did in this paper is, I said, they took metagenomic data sets. These are DNA sequences that have already been done and put in the database. And they looked at data sets from groundwater, sediment, acid mine, drainage, biofilms, soil, infant gut, and a bunch of others. They sorted through 155 million protein coding genes. 155 million, of course. This is bioinformatically or computational biology at its finest, because these repeating type elements are very difficult to assemble. Not easy to do that. So what did they find? They found the first Cas9 protein in archaea, and they found 2 brand new CRISPR-Cas systems in bacteria. They call it Cas-X and Cas9. So let me tell you a little bit about what they have done here. So the archaeal sequences, they reconstruct the genomes of 2 candidate archaea. One is called the Nanoarchaea candidatus microarchaeum acidifilum, which they abbreviate as ARMON1, and the other is candidatus parvarchaeum acidifilum ARMON4. And they put together CRISPR.
Can I interrupt for a second?
Yep.
I find this fascinating that not only do bacteria show reduced genomes, especially in endosymbionts, but they can find this in archaea. And at least one case, the archaeon has to live in symbiosis with another archaeon.
That's right.
So this is a general phenomenon that spans from bacteria to archaea, that they can lose a lot of their genes and still survive with the help of somebody else.
That's right. So they reconstructed 16 different CRISPR arrays. So a CRISPR array is, again, these repeat sequences, repeat spacer, repeat spacer next to a Cas, a couple of Cas genes, which are involved in acquisition and processing and immunity. And the one they found in Armon archaea, they found a lot of variability in the spacer content. Remember, the spacer is coming from incoming DNA. And so that makes them think that it's active in the sampled populations. The more diverse it is, the more likely it's still being active, because when you get infected, when you acquire new DNA, you're always making new spacers to take care of that. They also find this is very interesting, they have enough sequence from these spacers that they can sometimes reconstruct what the DNA looked like. And they have, in one case, a 10 kilobase pair sequence that they think is a virus. So they're pieces of the virus that they can put together and show that this is probably viral. And they say, yeah, we have seen virus particles in these cells, so it could be that. They find some evidence for transposable DNAs that probably move from cell to cell. And so they think that it's this Cas9, which they've found in the archaea, may have been against transposon propagation. Now, the other Armon4, the Cas9 is a little bit different. It only has one Cas9 gene. So it's not a typical CRISPR-Cas array. So they think it's probably not acquiring additional spacers. They don't know what the target for the spacer sequence is. It may have a single target and may have some other role besides defending against infections. Now, an important part of the CRISPR array is what's called a protospacer adjacent motif, or a PAM. It's right next to the target sequence. It's 2 to 4 bases long. It's very important because if you think about it, when you make a transcript from a CRISPR array, that's going to target DNA of the same sequence. What would prevent that from cleaving your own genomic DNA? Well, the answer is these PAM sequences are adjacent to the spacer. The transcript, the RNA transcript, will have the PAM sequence. And that sequence is not going to be found in the incoming DNA, so that DNA will get cleaved. But if the PAM sequence is found in DNA, the assumption is it's your own DNA, so that doesn't get cleaved. Isn't that amazing?
Well, it's necessary.
Yeah. Yes,
So,
Michael.
Vincent, is this Mother Nature's riff on restriction and modification systems that many of our listeners are already familiar with? The molecular scissors that effectively Herb Boyer pioneered with his description of EcoR1 back in the 70s, and this is just our version, but a much more sophisticated version of what EcoR1 does in E. coli, recognizing foreign DNA and then going after it. And the scissors, like EcoR1, is very specific in what it cuts, and these CRISPR-Cas sequences for these other organisms that they're pulling out of these sophisticated databases are just showing us that Mother Nature is constantly looking to improve its opportunities by doing experiments on its genome.
For sure. Here the target sequence is a little bit longer, but it's the same idea, right?
The specificity is better because target's longer.
Exactly. It's in each case it's the same protein, or there are a few proteins that are doing the cleaving, like Cas9, right? Which is a little different from the restriction enzymes, where each enzyme has its own recognition sequence. But this is brilliant. You have a nuclease protein and you stick a RNA onto it, which gives it specificity to a sequence. That's just brilliant, right?
Mm-hmm. Mother Nature is, if anything, brilliant.
Absolutely. Mother Nature is brilliant. I agree. A few more, a few more little bits here, and then I'll tell you about some experiments they actually did. So sometimes some of these CRISPR-Cas systems required two RNAs. The CRISPR RNA, and then there's a second one called a transactivating CRISPR RNA, and these have to hybridize in order to get cleavage. And they found these transactivating.
That sounds a bit complicated.
It's very complicated. The way the RNA actually hybridizes, so you have a double-stranded DNA, part of it melts, and the CRISPR RNA hybridizes to that, and then the second RNA hybridizes to one end of the CRISPR RNA. So you have three components. You have double-stranded DNA and two single-stranded RNAs in there, and that's needed for the cleavage system. So they find these transactivating RNAs in both of these archaea, and they may make this interesting.
Readers may want to refer to the notes, the show notes, because some of this is best seen in a diagram.
Yeah. So this paper, unfortunately, is closed access, but there's a wonderful CRISPR article on Wikipedia, which I'll link to, and you can see pictures of all this. And if you might want to look at it while we're talking, because it will help a lot. The diagrams are very clear there. Now, archaea were previously thought not to have type 2 CRISPR systems because they don't have RNase 3, which is an enzyme needed to mature the CRISPR RNAs.
Let me pipe in. This does not mean that archaea don't have CRISPRs. Au contraire. 90%, I think, of them have CRISPR, just not this particular system.
That's right, this particular
and the title,
system.
I think I found the title of the paper a little bit not misleading, but it could have been, it sounds like they found new CRISPR-Cas system from uncultivated microbes. Sound like these guys, like that's news in itself. What's new is the particular CRISPR systems that they found.
That's right.
Sorry to be picky about that.
No, it's right. So they found Cas9 in archaea, which hadn't been found there before. But as Elio says, archaea certainly have other CRISPRs. It's there, but it's going to be different because there are no RNase 3 in archaea. And then they found two new Cas proteins, which we'll talk about in bacteria. Now, they actually did a bunch of experiments here in this paper, which are really interesting. They reconstruct these and so in the case of the archaea, they actually make a DNA encoding this novel Cas9 protein, and they do this in E. coli and yeast to see if it has any targeting activity. So they give it an RNA, but they didn't find any activity of this Cas9 protein. So obviously they have to do some more work there. Something is missing. All right, now they have two bacteria recovered from groundwater and sediment, where they find some new Cas proteins. They have one called Cas-X and one called Cas9. And they did some experiments to see if Cas-X is capable of RNA-guided DNA targeting. So they make a plasmid encoding a minimal CRISPR-Cas X locus. They put a short repeat spacer array in there, and they put the spacer from a plasmid, which they then put into a cell, and they say, will this CRISPR-Cas X inhibit the introduction of this plasmid DNA? Okay, so the spacer matches the plasmid. They put their new CRISPR-Cas9 into E. coli, and then the plasmid has antibiotic resistance on it, and they simply grow the bacteria in the presence of antibiotic. And if the CRISPR-Cas is active, bacteria are not going to form colonies. And in fact, that they actually found that this minimal CRISPR-Cas X locus, which again they've identified in a DNA genomic set, is actually active. How about that? Isn't that cool? So that means they.
Why is it cool? Wait a minute.
It came out of a sequence. They just, oh look, we found this sequence. We make it. We put it in E. coli, and it works.
This is all in silico. They discern the sequence from the computer, then they synthesize the sequence, and voila, it worked.
And it is a brand new Cas.
That's pretty good. I agree.
It's a brand new Cas protein. It's never been seen before. They also identified a PAM sequence. Remember, a PAM is the adjacent couple of bases that gives it the ability to discriminate between foreign and host. And so what they do is they use this reconstructed CRISPR-Cas9, and they put randomized 4 bases next to the spacers, and they can then pull out the ones that actually don't cleave their own DNA and identify the PAM sequence for that particular one. So I thought that was pretty interesting as well. And they do some other experiments to show other parts of the CRISPR. For example, they want to know if there's a transacting RNA involvement, because there's some indication in the sequence that there is one. And in fact, when they delete the tracer RNA encoding sequence from their plasmid, it abolishes transformation interference. So basically, they have gone from the sequence to showing in a number of experiments that Cas9 is a functional DNA targeting dual RNA guided CRISPR associated protein. And they also have another one called Cas9. This is interesting. This is also part of a minimal CRISPR-Cas system. Again, they showed Cas9 is active. Cas9 is a separate one. There's no tracer RNA in this, and they weren't able to get activity from this one. So basically, now they've identified in these data sets Cas9 in archaea. But I want to tell you one more thing before I leave. Cas9, which is a minimal CRISPR, they synthesize the whole thing. They introduce it into E. coli on a plasmid vector. They put in the CRISPR, they put a sequence that matches the target plasmid, and they identify a PAM using this, and they get interference with the plasmid as well. So they have 2 new Cases, Cas9 and Y, which they show are active in targeting incoming DNA. And again, they identify them initially in a metagenomic data set. They reconstruct them in E. coli, and then they show that they work. And these organisms, as Elio said earlier, they're reduced to minimal genomes of many of these bacteria and archaea. And so we haven't been able to grow them, so we wouldn't have known about these CRISPR-Cas systems if we didn't look in the data sets. So they conclude discoveries related to CRISPR are not restricted to in silico observations, but can be introduced into an experimental setting to analyze their activity. And they make a nice statement. Given that virtually all environments where life exists can now be probed by metagenomic methods, we anticipate that the combined computational experimental approach will greatly expand the diversity of known CRISPR-Cas systems. And why that's interesting? Well, because we'd like to know what has evolved out there, but also we have, of course, started to use many of these CRISPR systems to, do engineering of various sorts, and we may find that there are more systems out there that are useful for us. Okay, Michael, what do you think of that?
I think this is absolutely fascinating. I had a tremendous sense of deja vu. Back in the early 2000s, we worked on hammerhead ribozymes, which are, again, RNA molecules that have a catalytic activity built into them. So you don't need the protein And again, you begin to see how these sophisticated editing tools are held on by the organisms because editing the genome is so critical for the selective pressure that they're under. I found it absolutely fascinating that the archaeans actually have this, and because the archaea are in this complex community, and it's to their selective advantage to edit down their DNA so that they don't have to make all those phosphodiester bonds. Right. And, it's absolutely fascinating and how it's done. But at the end of the day, it's natural selection that coordinates this. If they screw up, the microbe is dead.
That's right.
If it's a good phenotype, they're alive. And so it gets moved vertically through the genome. And again, these archaeons were named because they were thought to be old. They're the third kingdom. We have the bacteria, we have the archaea, and then we have the eukarya. And, those are effectively I don't know if we're still using the word kingdoms or they just call them bacteria, archaea, and eukarya. Domains. There's still domains.
Yeah,
if
domains
not, we'll get a letter from Norm Pace.
So I think the CRISPRs are really interesting. And as I said, if you find the discussion confusing, I'll put the link to the Wikipedia article in the show notes. And they have 3 good diagrams showing you the different kinds of CRISPR-Cas.
But I think people should be warned. It is complicated.
It is complicated.
Oh, you cannot really reduce it to just a couple of things. So just be aware of that. It's not the reader's fault. That's
not
the way
at all.
it is.
Not at all. No, we have type 1, type 2, and type 3 kinds of CRISPR. So they each have different mechanisms. And so
it's worth saying that it's worth going into it because CRISPRs are here to stay. They already have a lot of application in making mutations and inserting new sequences. They're really a basic tool that's going to be all over the place. So it's worth the effort. Let's put it that way.
So this is the concept that I equate this to the understanding of Apron topology that Elio wrote a blog post on this past week on Jacques Monod.
Mm-hmm.
And, it's absolutely fascinating because in the 50s and early 60s, operons were all the rage. And today they're taught in high school. And I imagine in 30 years, CRISPR is going to be taught in high schools.
Oh, for sure.
Because we will have
less than that.
We will have
less than that.
We have, systematized it, and I'm looking forward to. This is since I'm in LA, I can ask Hollywood to make an animation for us on how CRISPR might work, and a 3-dimensional model of how
that'd be
all
very
of this
nice.
happens.
If you could convince somebody to do it,
yeah, I might have been done already.
It would really be helpful.
Well, I looked and I couldn't find a good model on it, but
the problem is that there are 3 different mechanisms, and maybe they don't want to at least 3. There are probably more. So there are 2 classes of CRISPR-Cas types, class 1 and 2, and the 2 includes Cas9. Those are the ones with a single nuclease, and class 1 has multiple subunits to that nuclease. And then within each class, there are types, and the type 1, 2, and 3 are 3 different mechanisms, basically, that have been well studied. And there are different ways of producing the transcript, cleaving it, and targeting the incoming DNA. So you should check that out. It's a really good article. Okay, let's do a couple of emails here before we wrap up. First is from Saul, who writes, "Oh, I hope I win. I love your podcast. I listen to all these kinds of science podcasts like Are We There Yet?, Planetary Radio, Star Talk, Science Friday, etc. Happy to be included with those podcasts, Saul."
Nice.
Dale writes, "Hi, TWiM team. I'm a graduate student of medical microbiology in Port Elizabeth, South Africa. At the moment, I study antimicrobial resistance in hospital-acquired infections. I love listening to TWiM as I do my lab work. It keeps me updated, entertained, and hearing about all the amazing articles you discuss really inspires me to continue into research. I love books, and it would be awesome to add the color atlas to my growing library. Fingers crossed, and thank you for all the great content you produce."
Nice.
Sabrina writes, "Dear TWiM crew, I was so late listening to this podcast, so I know I missed the color atlas, but alas, I will try anyway. There's probably a new contest afoot. I am a clinical laboratory scientist in Central Florida, and your series of podcasts were actually what triggered me to go back to school to get my license to work in the clinical lab. Keep up the good work. So I love that kind of stuff.
Isn't that nice?
Isn't that great?
Isn't that nice? Thank you, Sabrina.
Melissa, hi, TWiM team. I can't remember if you've already discussed this video on TWiM, but Harvard Medical School released a time-lapse video by the Kishoni lab showing bacteria adapting to various concentrations of an antibiotic on a giant agar plate with different concentrations of antibiotic. I think
we did talk about it, didn't we?
I do think we talked about this. Yeah, it does sound familiar. We're all looking forward to listening to more of your podcasts. We, the Pride Lab, listen to your podcasts while we work. Melissa's at the University of California, San Diego.
The Pride Lab is a wonderful lab. They study phages in humans,
in
the human body. Lovely.
They're interested in the mouth too, right?
That's right. Yeah, they're all over the place. Beautiful work.
All right, we have one more email from a listener who is a frequent writer to our TWiV and TWiMs. She's a pediatrician in Cambridge, Massachusetts, and writes, the second paper about arbitrium, that was a couple of TWiMs ago, highlighted the gaps in my knowledge and left me with unanswered questions. In fact, I needed to re-listen to segments of the discussions multiple times to be clear about what I didn't understand. May I have a review of some terms and techniques? Making conditioned medium is a new term for me. Is it media that has bacteria grown in it? Yes, that's the answer, right? How long does bacteria need to be grown to be considered conditioned?
Gee, I'm sure it depends on the bacteria and the medium.
Yeah, I mean,
I don't think there's a general answer.
There's no answer. It could be 20 minutes or it could be several hours. And in the case of that paper...
Several days.
Several days. In the case of that paper, they were looking for something secreted into the medium that would signal other bacteria. And so that's one way you would use conditioned medium. What are some other uses for conditioned medium when you can use it to grow bacteria which may not grow otherwise, right?
I suppose. I don't really know.
Michael, what about...
I confess, I don't know.
We introduced this topic again here today when you were talking about transacting anything from food that a microbe may excrete as its waste product for another microbe, or it could be a quorum molecule. It's anything that a microbe throws away.
But how do you use it? What's the practical use for that?
Well, I think it's looking for things that are secreted by bacteria. In that case, it was the arbitrium peptide that influences other bacteria. It turns on signaling. Anything produced by bacteria, you could use conditioned medium. All it means is that the bacteria have grown in it. It may be a misleading term. All right, let me go on and finish this email. What is also the terms helix turn helix motif? Well, that is a motif in DNA. I'm sorry, it's a motif in a protein that's thought to bind DNA. And it just signifies that the protein has a helical structure, a turn, and then another helical structure, typical for wrapping around DNA. Chip sequencing. Chip sequencing is chromatin immunoprecipitation. You want to know what proteins are bound to DNA. So you take an antibody to chromatin, say, which are the histones bound to DNA. You can precipitate with that antibody. Then you can sequence the DNA to know what kind of DNA is binding your protein. Quorum sensing. Is it correct to think of it as describing a population's behavior based on its own density and resources available to support its viability?
Yeah, that's what it is.
Is that right?
Yeah.
So basically, bacteria make products that can be sensed by others, right? And they can reach a certain...
Of their own kind or even of different kinds.
That's right.
Usually of their own kind.
Hey, Michael, you there?
I'm here. Can you hear
good.
me?
Yep, we can hear you. We're almost done, Michael. Hang on.
Okay.
Hope my knowledge gaps are not too basic and elementary. I really became concerned when Professor Schmidt suggested there was a good follow-up project appropriate for high school. Or was that meant for the first paper? As always, warmest wishes, especially at this time of year on the East Coast, and many thanks, Johnny. All right. So we hope we explained some of those to you, Johnny. This has been TWiM 148. You can find it at iTunes, microbe.tv/TWiM, also asm.org/TWiM. And you can also grab it on your smartphone or tablet using apps that can subscribe to podcasts. Consider becoming a patron of TWiM and all the other Microbe TV family of science shows. Go to microbe.tv/contribute for all the different ways you can help us out. And, of course, send us your questions and comments to twim@microbe.tv. Elio Schaechter can be found at Small Things Considered. Thank you, Elio.
My pleasure, of course.
In the company of mushrooms. You know, Neanderthals are in the company of mushrooms, too.
Hey, smart people.
Michael Schmidt is usually at the Medical University of South Carolina. Today, he's at Los Angeles International Airport. Thank you so much, Michael, for spending time there to talk with us.
I really enjoyed it. Now I have to get on the famous LA freeways.
Well, Michael, have a safe trip and enjoy your meeting.
Oh, thank you very much.
I'm Vincent Racaniello. You can find me at virology.ws. I want to thank the American Society for Microbiology for their support of TWiM, Chris Condian and Ray Ortega for their technical help. Also, thanks to the sponsors of this episode, Blue Apron. The music you hear on TWiM is composed and performed by Ronald Jenkins. You can find his work at ronaldjenkins.com. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.